Distinguished guests and esteemed colleagues, I am delighted to welcome you all to the fifth session of Cerrahpasa Neuroscience Talks. Today we have the great privilege of hosting an exceptional scholar and influential expert in the field of neuroscience and behavior, Professor Dr. Nils Kroemer. Our focus today will be on an intriguing topic that lies at the core of our understanding of human behavior and cognition: vagal vitality, brain-body interactions in the control of goal-directed behavior. As we eagerly await the insightful talk by Professor Dr. Nils Kroemer, let us first acquaint ourselves with his illustrious background and accomplishments in the realm of neuroscience. Professor Dr. Nils Kroemer studied psychology at TU Chemnitz and obtained his PhD from the psychology department of TU Dresden in 2013. During his postdoc, he also studied at the John B. Pierce Laboratory, Yale University. He is now the head of the Section of Medical Psychology at University of Bonn and group leader in the Computational Psychiatry of the Section of Translational Psychiatry at University of Tübingen. Professor Dr. Nils Kroemer is the principal investigator of the neuroMADLAB. The work of his lab focuses on the neurobiological principles of motivation, action, and desire. He has published over 50 peer-reviewed articles and has received several awards and recognitions for his work, including the Werner Straub Award for his outstanding PhD thesis and an IZKF Junior Research Group Award. It is an immense privilege to have Professor Dr. Nils Kroemer as our esteemed guest, and we eagerly anticipate the wealth of knowledge and expertise he will share with us today. After his presentation, we will have a Q&A session where you can type in your questions in the chat box or address them yourself. So, without further ado, I welcome Professor Dr. Nils Kroemer, and I am looking forward to him to enlighten us on the captivating topic, vagal vitality, brain-body interactions in the control of goal-directed behavior. Professor, welcome. You have the word.
Well, thank you very much for the very kind introduction, and thanks again for having me. I'll share my screen in a second, and then we'll be good to go. All right. Today, I'll try to walk you through some of the work that we have done in the past few years. And I'll focus in today's talk on brain-body interactions and how they help us to basically adjust goal-directed behavior according to metabolic state, right? And what I want you to take away from today's talk is that the vagus nerve plays an important role in adjusting goal-directed behavior, and that we have the opportunity to target the circuit using non-invasive brain stimulation. And we can also use those tools to learn something about how basically our body helps to guide behavior, right? So let's get started. And first of all, I want you to just think about think for a second about eating as a specific kind of instance of value-based decision-making, right? So we have an intuitive understanding of eating, and we don't necessarily have to conceptualize it as value-based decision-making. But it's useful for us as neuroscientists to think about that sort of framework, because it may help us to see some of the mechanisms that can also be useful, not only to understand how we decide which meal we want to eat next, but also maybe some of the processes that come into play when we think about mental disorders, right? When we think about lack of appetite and changes in body weight during major depressive disorder, but also when we think about eating disorders, some of those derailed processes that we're still trying to target more specifically with treatments. So if you think about the typical kind of choice, we have to select from a set of options. And, you know, if we look at the modern industrialized countries, we often have more than enough food. So the challenge is not to find sources of food, but to select from a large set of potential options. How do we go about this? So we basically have to consider different dimensions that characterize each potential food item, right? And that's a very difficult kind of decision. So we usually have to narrow it down and simplify the kind of process, because the potential options are unlimited. And what we use to guide our choices is basically we tend to use tastiness as a sort of proxy. Tastiness is something that tells us usually how rich a certain food is, right? It is closely associated with caloric density of the food. But we may also take other dimensions into account, such as how healthy is the food option, or how sustainable is the kind of food that we consume. That's something that is certainly a growing concern here in Germany as well. And then we have to make choices, and we also have to make some sort of instrumental response, right? An instrumental response means it's a goal-directed behavior, right? And we may have to walk somewhere to get the food, or wait in line to enter a favorite restaurant. Those are kind of instrumental responses that may come with a given cost, right? Effort, investment of time. And those instrumental responses are therefore also a very good proxy of how much we value certain options. If you think about typical choices, right? Effort is usually associated with how much you value a given outcome, or also how long you're willing to wait in line for, say, your favorite food is a good proxy of how much you value that particular food item. Now, something that we're interested in is also, how does flavor shape our choices and instrumental responses? What are some of the psychological effects of food? That sounds, more cryptic than it is. Psychological effects of food are something like changes in mood after we had our favorite meal. I'm sure you're familiar with the concept of comfort food, right? Something that we may seek when we feel down, and that may brighten our mood to some extent. And one thing that I will focus on in today's talk is also the role of post-ingestive signals in helping us guide our future choices, which is also related to memory processes, right? So we do have vivid memories of where we had a good meal. I could still recall some of the instances where I was traveling and had a very good meal when I was abroad, but also within Germany. And the way it works is basically when we consume food, right? We do have a certain idea how rich that food is in terms of calories. But then we update our expectations that are dependent on, flavor as something that we use as information, with extracted energy once the food is broken down in the digestive tract, right? And that's what we refer to as post-ingestive signals. And part of that information is forwarded via the vagus nerve to the brain. And this helps us shape preferences for rich food as well. But I'll go into this in more detail in a minute. And something that we have to consider as well, that there are inter-individual differences. We all know about the variability in terms of body weight and the growing obesity epidemic. And it's not like if we were in the same situation and had the same set of options, we would pick exactly the same kind of food, right? We're also flexible and dependent on the context of in terms of how we make up our mind on a given day. So it's a relatively complex scenario. And the question is, how do we sort of optimize our behavior, okay? Now, this is a simplified way to think about how we make inferences about food. And on the one hand, here you can see, let me get the laser pointer. Here you can see so-called higher order cortical areas that are involved in food choices. And they are basically supporting conscious evaluation of food. So when we consume a given food like a strawberry, we can name that food item, and we can also, sense it with a distinct flavor profile. And that's all fine, right? We tend to think that a lot of our food choices are driven by those higher order cortical areas and very deliberate. But we tend to ignore a more subconscious element, right? Those are the subliminal reward signals that are forwarded from the gut, which I refer to also as post-ingestive signals before. And this pathway is important to basically ensure survival of the organism, right? It basically is a form of hardwired circuit to make us prefer food that is rich in carbohydrates or fat, so very energy dense. And we can also put this into more computational or decision-making terms. We tend to refer to the input from higher order cortical areas also as model-based control or reflective behavior, right? This is when we inhibit, for example, a prepotent response to something that is rich in calories and go for something that is a healthier option instead. Whereas we characterize the subconscious element more as a reflexive kind of approach or a model-free control. We don't really have to give it much thought, okay? So this is our initial preference usually. And the way we conceptualize this kind of trade-off between those two kind of pathways to desire is that we can use reflective control on top of this kind of reflexive mode. But this is dependent on limited resources, right? So for example, when we're distracted, when we're stressed, when we're in a negative mood state, we're less likely to exert model-based control because we do see it as costly process. And then we'll more likely go for the kind of comfort food that we also crave, right? And that's one way that we try to conceptualize also eating in terms of value-based decision-making and link it to some of the interoceptive signals that we're using to track the effects of food inside our body. Now, I'll focus more on what the vagus nerve does in that sort of integration, right? Why can we now target the vagus nerve? And that's actually something that is, very helpful for practical reasons as well. There is an auricular branch of the vagus nerve that we can target with a simple electrode, right? And here you can see basically where we have the auricular branch of the vagus nerve. And those are not exactly the same kind of fibers that come from the gut and are connecting to the brainstem. But we do know that this auricular branch of the vagus nerve also basically projects to a target region in the brainstem here that is important for relaying all sorts of interoceptive signals to the brain. Interoceptive signals, basically everything that is a bodily signal. And the big question that we had in the beginning was, if we stimulate the vagus nerve at the ear, can we alter brain responses? Can we also have afferent effects on organs of the body? Can we basically trigger that entire feedback loop so that we can elicit changes in behavior? Because at that point, there was a different focus within the human community as well. And there wasn't so much work trying to connect the more physiological work in animals to non-invasive vagus nerve stimulation in humans. Now, a little bit more about this gut-brain interface that I've been talking about already, right? If we try to put it into more physiological terms, on the left side, you see a very simplified kind of scheme where you see vagal afferent projections to this small nucleus in the brainstem called the nucleus of the solitary tract, or NTS. And there are known projections to the ventrotegmental area and the substantia nigra, which are some of the key regions of the dopaminergic midbrain. And then there are projections also to the striatum. Now, what happens is shown here with the rodent eating the ice cream, right? It basically is activated once calories are detected also in the gut, and it basically gives a delayed feedback signal to inform us that this is a valuable source of energy. Okay, and this leads to a dopamine release also in the striatum that can condition place preferences and flavor preferences. This is something that has been shown in the Cell paper in 2018, also by the same lab, Ivan Darujo's lab. They basically showed that if you stimulate the right nodose ganglion, you can condition flavors, you can condition preferences, and that animals will also work for stimulation of the right nodose ganglion because it is inherently rewarding. Okay, and all of that is dependent on the release of dopamine in the midbrain and the striatum. What is interesting as well, and was a big puzzle until recently, is why would you see such motivational effects of vagus nerve stimulation when we think that most of the information that is conveyed by vagal afferents is actually related to satiety? So when we started looking into vagus nerve stimulation, this is where you could read that most of the information forwarded by vagal afferents is basically a negative feedback signal, okay? So it will likely reduce motivation or suppress food intake. That was the understanding early on, but it has been shown as well that there are parallel neuropopulations in that small brainstem nucleus, the NTS, that can either increase or decrease food intake. So it's also possible that, depending on the kind of stimulation of vagal afferents, that they would trigger different kind of neural cascades that can increase food intake or decrease food intake, depending probably also on contextual aspects. But vagus nerve stimulation is also more broadly involved in memory formation and food seeking. As I said, that you remember where you had a good meal is probably also related to vagal afferent signaling, right? Because it's something that is delayed, usually when we use those post-ingestive signals. And then it has been shown that this process can also help us store those memories. So just a quick rundown on how non-invasive vagus nerve stimulation works. So 20 years ago, the head of the psychiatry department in Tübingen actually showed that if you put an electrode at the cymbal concha and you stimulate what was thought to be the auricular branch of the vagus nerve, you can indeed induce so-called far-field potentials, indicating that you stimulate the brainstem, even though the resolution of deep neurostructures with EEG is not very good. But that was the earliest kind of indication that it might be possible to stimulate at the ear and to elicit changes in brain responses that are in line with an effective stimulation of vagal afferents and the brainstem. This has then inspired the design of a device that we use as well, which is approved for various medical applications as well. And the way we use it is, very similar to 20 years ago, but the electrode was optimized so that it's easier to position the electrode. And we are also using a sham condition that you can see here on the right side, where we put the electrode upside down. And then we match the sham condition to the actual stimulation of the vagus nerve in terms of sensory effects. So we basically have participants rate on a visual analog scale their pain sensation, how intense they feel the stimulation, and then we can match the two stimulation intensities so they feel about the same. And it has been shown in earlier work that you have to go below the pain threshold to elicit robust brain signals as well. So we're doing that for both of the conditions. It doesn't feel very rewarding in humans with the kind of protocols that we're using. So it's not really comparable to the invasive stimulation of the right nodus ganglion where people would work for the stimulation. But, it is also a different kind of stimulation protocol after all, and it might be possible also to optimize those aspects in humans. But I'll get back to this by the end of the talk, that this is something that we're still trying to figure out. Now, the big advantage of the setup is we can do tasks in parallel, and I'll walk you through some of the findings, but you can also do functional neuroimaging in parallel. So while participants are inside the scanner, we can stimulate the vagus nerve, we can measure in real time the effects on the brain, and then we can see how changes in brain responses may also explain some of the changes in behavior or correspondence with bodily signals. You may wonder why this small nucleus in the brainstem is so important, right? So it's a small nucleus, but it's very well connected to a lot of important regions within the brainstem and the midbrain that basically regulate, neurotransmission across the entire brain. So you cannot really say that vagus nerve stimulation is a selective method that alters only one neurotransmission system, right? Even though it's very focal, it has a widespread effect on neurotransmission throughout the cortex. And that's something that is often desirable, specifically for treatments of mental disorders. We usually have to target multiple neurotransmission systems at the same time. And it just indicates also the privileged position of the brainstem, right, in regulating something like, arousal through modulation of cortical activation. And that's, partly done by triggering noradrenaline release in the locus coeruleus, and then also upregulating release of acetylcholine throughout the entire cortex. And this plays an important role in motor learning and potentially also some of the motivational effects that we'll talk about. But of course, one idea is also that part of the motivational effect is driven by dopaminergic projections, although that's very difficult to isolate from noradrenergic effects by just looking at behavior. First of all, when we look at some of the data now, I want to make one point clear, and that's we can indeed stimulate the brainstem effectively with this method. So that's our one kind of reassurance that the method works as intended. And this has been shown across many different labs with various kind of setups as well. Just to give you an idea, those are three studies that looked at changes in brain responses within the brainstem in response to vagus nerve stimulation. And we also completed a so-called registered report where we submitted our experimental protocol in advance for review, and then it was already approved in principle. And then we collected the data, and then we ran all of the analyses that we had pre-registered. And this also indicated that we see a robust increase in brain response after vagus nerve stimulation in line with previous studies. So this is reassuring in the sense that we can activate the brainstem. And I think that's like the prerequisite for most of the behavioral effects that we're also interested in. I do have to say, though, that it's clear from the data that we have collected that we're not increasing brain responses in every individual as well, right? So there are even some non-responders to this kind of stimulation when we look at, the closest kind of outcome that you can imagine in terms of, immediate effects of the stimulation. So that's something to take into account, that there are likely also people included who are not showing an activation of the brainstem, and that might be dependent just on how their auricular branch runs through the ear and whether we can effectively stimulate it at the surface. Now, let's move on to the results from our motivational paradigm. And here I just want to give you an idea of effort and motivation in the wild. So here you can see the small rodent trying to carry some food through the hole. And something that you can see in this little video is that there are two components that you can isolate from this sort of behavior, right? And one is more the kind of invigoration, the jump that the little rodent has to make to get through the hole, and the other element is effort maintenance or perseverance. Okay, so you have to keep going if you want to make it often, and we can use both of those components also as proxies of value, how much we value a given thing. We have implemented a task that captures this sort of process. And we asked our participants to come into the lab after an overnight fast. So they were very hungry and motivated to work. And we told them, look, this is your task. You can work for your breakfast now, okay? And we then gave them an Xbox controller, and we asked them to repeatedly press the right button on the controller. So they saw this blue ball.
Kimle konuşuyordun?
They
Aşağıdan
had the option to move this blue ball above the red difficulty line. Okay. And the more often they press the button, the higher this ball would move, right? So their job was to basically keep the ball above the difficulty line so they could collect rewards. And we used either food rewards or monetary rewards, and we manipulated also how much they could win in terms of reward magnitude. And we also manipulated the height of the line here to make it either easy or hard to get the rewards. Now, this is a summary of the task design, and what we got from the behavioral data was, as I said, two different phases. Invigoration slope is basically how quickly people ramp up their behavior to collect the rewards. And then effort maintenance is related to how much effort they produce over time. And we didn't see an effect of vagus nerve stimulation on effort maintenance. This was just a trend. And for this invigoration phase, we did see an increase after vagus nerve stimulation. And if we stimulated the left ear, it was primarily linked to food rewards, the effect. Whereas if we stimulated the right ear, it generalized beyond food rewards to monetary rewards. Now, one of the questions that I also received was related to those differences in lateralization. And one idea is that when you stimulate the right vagus nerve, you are more likely to elicit a strong dopaminergic signal. And we think that the best explanation is that some of the food-related processes are dependent on vagal efferents, independent of dopaminergic signals probably, or somewhat independent of dopaminergic signals. But if you want to go beyond food rewards, you will probably also have to stimulate the right vagus nerve. Okay, so it's not entirely clear in humans whether left vagus nerve stimulation can also elicit a dopamine release. But at least in rodents, it's quite clear that there is a substantial difference between the left and the right branch of the vagus nerve. And we also collected subjective ratings of wanting. And remember that I said that those signals from the gut are largely subconscious in driving our behavior. So we also looked at conscious ratings of wanting that were collected after each trial. And we didn't see an effect on subjective ratings of wanting. So what essentially happened is something that you can see in those plots. In the conditions where vagus nerve stimulation increased invigoration, you see that the correspondence between wanting ratings on the x-axis and invigoration on the y-axis was reduced with vagus nerve stimulation, indicating that participants worked harder in a sense, or wanted to get the rewards more that they actually rated as, not so interesting in terms of wanting. And that's something that we interpreted as an indication of motivational drive, right? Motivational drive is somewhat disconnected from the conscious report of wanting. It's more automatic. And that seems to be something that is altered by non-invasive vagus nerve stimulation, in line with the idea that this is more of a subconscious automatic kind of approach. We then ran also some permutation tests to show that this is a robust effect, and we have also replicated this sort of change in independent samples by now. I also mentioned briefly that this is of interest for applications. We heard this also in the introduction, that we're trying to develop, in a sense, translational approaches to modify reward-related behavior. So I want to share with you some data that we're currently collecting on differences between patients with major depressive disorder and healthy control participants. So they did the same kind of task without vagus nerve stimulation in this case. And something that we see here is patients with major depressive disorder, they tend to work even more for the low rewards, whereas they work less for the high rewards. There's not so much of a difference between easy and hard trials in terms of behavior. And when you look at subjective ratings, it's also similar that the difference between high and low reward magnitude is smaller, indicating that they have a lower reward sensitivity that drives the differentiation between high and low reward magnitude trials. And that's something that kind of surprised us to some extent. It has been shown before that patients with major depressive disorder have a slightly lower reward sensitivity, but we were thinking that maybe they would be less motivated across the board to work for rewards, or maybe also show lower ratings of wanting across the board, but that was not so much the case. But what was really striking in terms of changes was that patients with major depressive disorder showed a weaker correspondence between their subjective ratings of wanting and how much effort they invested on the task. So this is something that you can see here. If you compare the blue line for patients with depression to the yellow line or golden line, then you can see there's a reduced correspondence again by wanting, between wanting and effort on a given trial. So one thing that we take away from the kind of data is maybe that there's also an alteration between how patients with major depressive disorder integrate, reward-related information over time, that there is a discrepancy between behavior and conscious ratings of wanting. And one idea is that this is also due to, this connection between interoceptive signals and exteroception, something that they learn about the world and how they process the world. But that's something that we're looking into now. But at least this data is pointing also towards an alteration in how conscious evaluation is aligned with motivational behavior in major depressive disorder, something that we can perhaps target with non-invasive vagus nerve stimulation. We have also collected across other tasks subjective ratings, and I have to say this, based on a lot of data, we can show that it doesn't really change subjective ratings of wanting or liking. If you look at healthy participants, but we did see that vagus nerve stimulation improved food liking in patients with major depressive disorder, and this was primarily driven by patients who suffered from a loss of pleasure. And then we did some additional analyses to show that you may describe also the effects of vagus nerve stimulation as something that has more of a normalizing effect on ratings. So it doesn't just increase or decrease ratings across the board, but if you have lower ratings, it may also improve them to some extent and, perhaps support a reversal to a more normal state, something that we interpret as potentially an increase in reliance on interoceptive signals as well within major depressive disorder. But that's something that we're following up on now. So this is more of like a initial study to see whether it is promising as well to target this in major depressive disorder. And before I'll do a quick interim summary, I also want to show you that we observed acute effects on mood. This has been reported before. And what is interesting about the positive effects on mood is that they occur after we stop the stimulation and not while we apply the stimulation. So this is something that you can see here. This is data in healthy participants. You don't see much effect on negative mood, which is low in general. But positive mood decreases when they have to, exert themselves. They do physical effort tasks, more like cognitive effort tasks as well. And then the positive mood decreases. And then they get the rewards that they have earned during this recovery period. And then what we see is even if you control for how much they win, after vagus nerve stimulation, they see a much more a facilitated recovery of their mood after the stimulation has ended. And we also ran some statistical models to demonstrate that this effect is specific to this recovery period and does not occur already during the acute stimulation phase. So it's also very pronounced in that later phase. But that's also something that you want to see for a potential treatment of major depressive disorder, right? So you don't necessarily want to just get motivational effects. You also want to get improvements in mood if possible. The interesting thing is that they don't seem to occur at the same kind of time scale, right? You see acute effects on motivation, but not acute effects in terms of mood. They seem to occur after the stimulation period. Okay, just a quick summary. Vagal afferent stimulation boosts motivational drive. We do see robust TVNS-induced increases in brain responses in the nucleus of the solitary tract. And we see increased invigoration to work for rewards. We do also see altered reward learning. I haven't included this kind of data, so you have to trust me on that. This is basically in line with hypothesized changes in monoaminergic tone, so primarily changes in noradrenaline and dopamine. And we also see improved mood recovery after challenges, and we don't see changes in subjective ratings of wanting and liking in healthy participant, but we do see increases in patients with major depressive disorder and anhedonia. Now, I think the big question is, what does that have to do now with, effects on peripheral organs, and what does that have to do with interoception? I've been trying to hint at some of our interpretation already, but I haven't showed you the data yet, but we think that this is due to altered interoceptive signaling as well. Now, for you to understand why it makes sense to assume that we can also alter efferent signals, I have to show you this kind of circuit, right? So far, I've been talking primarily about vagal efferents, and if we stimulate them, we can trigger a response in the nucleus of the solitary tract. But the good thing is that we know that there are also projections from the nucleus of the solitary tract to the dorsal motor nucleus of the vagus nerve. And if we stimulate this part as well, then we should see efferent effects on, for example, the speed of the digestive tract, okay? Because that's controlled by vagal efferents as well. And if we're interested in that, we can actually use a very simple technique called electrogastrogram, which is like an electrocardiogram, but you put the electrodes above the area where the stomach should be, okay? And then you can collect with those cutaneous electrodes the, intrinsic rhythm of the stomach, which is about 3 cycles per minute, roughly. Now, if we turn on the stimulation, that's one of the earlier studies that we did in our lab, you can see that you can shift the speed of the digestive tract as measured by the frequency of gastric myoelectric activity. And this is depicted here on the top right. And basically in parallel to our study, there was also an independent report by a group that did invasive recordings where they put electrodes during a surgery on the stomach and used non-invasive vagus nerve stimulation. And they also found that vagus nerve stimulation decreased the speed of the digestive tract. So this is sort of an independent indication of the same kind of effect of non-invasive stimulation. Now, one reason why we were intrigued by this finding was also that there was another paper indicating that we have a resting brain network that basically is coupled to this intrinsic rhythm of the stomach, okay? So we can collect those electrophysiological responses of the stomach over time. And we can also do functional magnetic resonance imaging in parallel. And then we can look at how strongly those signals are coupled, okay? And this is what Ignacio Rebolo did in his eLife paper in 2018. And they identified a large brain network that is phase synchronized with the stomach. They did a lot of tests to ensure that this was not just an artifact, and they basically showed that this is related to somatosensory regions, primarily unimodal cortical areas. What's not really known, or what was very difficult to establish, is what the functional role of the gastric network, right? Because you can observe it, you can look at the correspondence with signals of the gut, but you don't really know what the functional role of this gastric network is unless you have a way to perturb this network as well. So we reasoned that if we can alter the speed of the digestive tract when we stimulate vagal afferents, it might also be possible to look at changes in the gastric network as an indication of altered stomach-brain coupling, basically altering gut-brain signals with a controlled stimulation. So we did the exact same setup. And first of all, we were able also to replicate the gastric network in principle. And then we looked at the changes that we could induce with non-invasive vagus nerve stimulation. And indeed, we saw increased stomach-brain coupling in the nucleus of the solitary tract and also in the dopaminergic midbrain. And actually across the cortex, we saw that in regions where there was stronger stomach-brain coupling to begin with during an independent baseline scan, we saw an increased after vagus nerve stimulation and stomach-brain coupling. So this was also associated across the cortex, indicating that what we can do is basically strengthen the correspondence between signals of the stomach and the brain. Okay, so we thought this was very nice. And then a few months after our study, we saw another paper in rodents, basically looking at the other kind of way to go about this kind of problem. They used vagotomized animals as well and studied the effects of stomach-brain coupling. So vagotomy is basically when you cut afferent and efferent projections between the vagus nerve and the brain. And you can see this here. They also looked at comparison between fat and fasted state and then the vagotomy condition. And what they found was if you compare a fat state to a fat state after vagotomy, you see that most of the stomach-brain coupling is actually gone, right? So this indicates that if you cut projections between the brain and the stomach via the vagus nerve, that you can also abolish stomach-brain coupling. So I think those two studies really indicate that communication between the stomach and the brain and this kind of synchronization is dependent on the vagus nerve. And we can alter it with vagal afferent stimulation, but we can also sort of lose those connections when we cut the projections back to the peripheral organs. And in the remainder of the talk, I'll show you some of the more recent data that we have also collected. So we also use the food bidding task inside the scanner to study basically how vagus nerve stimulation alters brain responses to food. And first of all, we were able to replicate the increase in invigoration with vagus nerve stimulation in a different kind of task in an independent data set. And again, it generalized to non-food rewards as well, and it was not specific to food rewards. Again, right-sided vagus nerve stimulation. And what was interesting as well is when we look at the behavioral changes in the food bidding task, right? So how much people are willing to bid to gain access to those rewards, we can actually do quite well to predict individual changes in behavior using brain responses to vagus nerve stimulation. And one thing that we tried is, can we actually predict those behavioral changes by just using the acute effects of the stimulation? So here we're still looking at a resting state scan before they do the food bidding task. And there are a couple of regions in the brain where we see increases in brain responses to vagus nerve stimulation, including the nucleus of the solitary tract. We also see it in the caudate and the bad nucleus of the stria terminalis and the hypothalamus. But there are even more regions where vagus nerve stimulation makes individual brain responses more variable in the sense that it increases for some people and it goes down for other people. I mentioned that this is likely due to some differences in anatomy of individuals as well. And one of those regions is, for example, the nucleus accumbens, right, where we see an increase in brain responses for some individuals, but also a decrease for other individuals. But in principle, vagus nerve stimulation increases how variable brain responses are in the accumbens compared to a sham stimulation. And we use those kind of regions where we can elicit inter-individual differences as well to predict behavioral changes. And this works quite well, indicating that those individual differences that are elicited by vagus nerve stimulation are likely responsible for changes in behavior as well. Okay, so the idea is that we could potentially use also acute changes in brain responses as something that tells us whether a participant is likely to benefit from the application of vagus nerve stimulation in terms of, motivational effects or also antidepressive effects, perhaps more broadly. But that's something that we're working on right now. This is like a preliminary analysis of the idea. I also showed you in the beginning this scheme where we're comparing higher order cortical areas to striatal and midbrain circuits that are more involved in subconscious processes. And that's something that we try to test with our data as well. So remember that we had the food cues inside the scanner, and we still collected an electrogastrogram while we showed the food cues. So one thing that we could do now is we could do this kind of phase locking analysis that we do for signals of the stomach, and do the same thing for the pictures that we show inside the scanner, and then compare if brain signals are more strongly aligned with the pictures or with the signals of the stomach. So that's the idea, that we can compare images, processing of images to processing of or synchronization with signals of the stomach. And if we do that, we see this very nice differentiation between cortical signals, right, of the visual cortex and the motor cortex that are more involved in controlling than also the behavioral response. And those regions in the striatum and the midbrain and the brainstem that even though we're showing images of food, those regions are still more strongly coupled with the rhythm of the stomach compared to the rhythm that is introduced by the food pictures. And that's also quite strong effect that we can see in those regions. So I think this speaks for the idea of those two pathways in the brain that are, on the one hand, more strongly guided by interoceptive signals. Those are the regions in blue, whereas we have also this representation of what is happening outside of our body, in our environment, in cortical areas. And then this has to be integrated likely in regions such as the ventromedial prefrontal cortex and the striatum then to some extent. Okay. And in the last part of the talk, I'll just briefly talk about how this relates also to subtypes of depression. I mentioned that in depression, there are also changes in appetite and weight. So we refer to those changes as subtypes also in depression. There is so-called atypical depression with increases in appetite and weight, and melancholic depression with decreases in appetite and weight. And something that we try to understand is if this is also aligned with changes in functional connectivity of the reward circuit, because the idea is that we can then potentially use techniques such as vagus nerve stimulation to treat some of the changes in appetite as well in a more targeted manner. And first of all, we looked at associations of functional connectivity of the nucleus accumbens, one of the main hubs of the reward circuit, with body mass index. And that was very similar for patients and controls. It was primarily associated with a weaker functional connectivity between the hypothalamus and the reward circuit, which I think makes sense because it indicates that there is a lower functional coupling between the homeostatic network and the reward circuit. And then we did an analysis of those kind of opposing changes in appetite and weight in major depressive disorder. And we also found that it was associated with changes in functional connectivity of the nucleus accumbens to the ventromedial prefrontal cortex, to the hippocampus, but also to the primary ingestive cortex in the insula, right? So we're basically getting a network that we also see in a lot of those studies involving food rewards. indicating that whether you see an increase or a decrease of appetite during a depressive episode is to some extent dependent on functional connectivity of the reward circuit. And the interesting thing for me, at least, was this was a very large sample of patients and controls. Up to 807 participants were included in this neuroimaging study at this point. And yet, if we wanted to classify individuals with depression versus healthy controls using functional connectivity of the reward circuit, it was not possible. It was only when we differentiated between increases and decreases in appetite, which to me are also very different changes in motivation in major depressive disorder. And then we were able to classify and predict basically changes in behavior. Now, I'll just take 2 minutes before I wrap up to talk about some of the future avenues, some of the open questions in the field. One of the open questions is, how does it relate to heart rate variability, right? You might have heard about vagal tone before and heart rate variability as a measure of vagal tone, but it doesn't seem to work very well. If we stimulate vagal efferents, we don't see conclusive changes in heart rate variability. So this is still something that is a bit puzzling. And we're also monitoring the evidence here and collecting more data. But one of the reasons is probably that heart rate variability, as we usually compute it, is not a very good measure of vagal tone, unfortunately. So that's a tricky part. Another thing that I've mentioned very briefly is a lot of people are interested in, eliciting cortical arousal, and we use pupil dilation also as a measure of noradrenergic effects of the stimulation. For a long time, it was puzzling why we didn't really see good changes in pupil dilation in response to vagus nerve stimulation. But there are some new studies out now using different stimulation protocols with shorter periods of stimulation, and they seem to work very well in inducing pupil dilation, indicating that it's possible to modulate noradrenaline release as well. So that's also encouraging that, there's the opportunity to optimize also the stimulation protocols and to elicit different effects on the brain. And of course, we would love to combine also vagus nerve stimulation with, a more tailored approach where we record signals from the stomach, for example, indicating hunger or something like this, so that we could modulate vagal signals to, for example, improve diets. This is something that has been done in rodents in a closed loop brain stimulation setting, but this is still a couple of years ahead in humans so far. I've also included this slide, but I will keep it very brief. I mentioned that vagal projections are also quite variable across participants, so there's also ongoing work trying to get a better idea of how fibers of the vagus nerve are going through our bodies, and it's surprisingly variable how they are, merging and splitting. So it's kind of difficult also for invasive vagus nerve stimulation to identify the right fiber bundle that should be stimulated, and that's still one of the main challenges. And we need to get better imaging techniques as well to measure those individual differences. And of course, we're interested in targeting, gut-based feedback signals more broadly. So we're also looking into ghrelin and GLP-1 as potential treatment targets, not only for major depressive disorder, but also for eating disorders and obesity. And that also seems quite promising so far. And we're also running studies using, for example, the infusion of ghrelin to induce motivation, with the long-term goal to perhaps normalize reward function in patients who suffer from anhedonia and major depressive disorder. Okay, and now, as I said, we're also running some follow-up studies in a larger group of patients with major depressive disorder, where we try to improve interoceptive signaling again, and then use that to improve somatic symptoms in depression as well. Now, this is the final slide. Vagal afferent stimulation is a promising method to subconsciously alter behavior and brain signals. We do see robust activation of the projection target in the brain, the nucleus of the solitary tract. We see increased invigoration to work for rewards, improved mood recovery, and we see altered coupling between the stomach and the brain, and also changes in gastric frequency. All those effects indicate that we can indeed modulate a vagal-vagal feedback circuit. We see some early indication of a good prediction of TVNS-induced changes in motivation by individual so-called stimulation fingerprints, so individual effects of the stimulation. And this may help us identify who is likely to benefit from vagus nerve stimulation. But we still don't really know how all of that, is orchestrated across the brain to guide behavior. So if we wanted to use it at a larger scale to help people, maybe improve their behavior and improve their goal-directed behavior more broadly, to lose weight or whatever, we would still have to optimize how we go about this. And there's a large need for innovation to understand how to achieve this. But I think it's promising that we can shape motivational drive and perhaps also measures of energy metabolism. And I think it could be very helpful to target interoceptive signals in the future. And with that, I want to thank, of course, the team in Tübingen and Bonn, and thank all of our funders, of course, and you for having me today. And I would be happy to take your questions now. Thank you very much.
Thank you so much, Professor, for your valuable presentation. We will now begin the Q&A session, and as Professor Dr. Kroemer told me before, we would love it to be as interactive as possible. So please feel free to type your questions or contributions in the chat box, and we will try to address as many of them as possible during our interactive Q&A session. You can also use the hand raise option as Efe is using right now if you want to address the question yourself. Actually, before we begin, I want to ask a question myself, if that's okay. My question goes back to your publication in Nature Communications, and it's a little bit specific. Your study found that the side of stimulation, so left or right, affected the generalization of the invigorating effect of transcutaneous vagal nerve stimulation. So can you explain the possible reasons behind this lateralization effect and its implications for understanding the neural mechanisms involved?
Yeah, so, it's kind of surprising that something as basic as a motivational effect of vagal afferent stimulation would be strongly lateralized, from an optimal design perspective, maybe. But it seems to be one of the peculiarities of how our anatomy has been selected for, right? So it starts in rodents that there is this lateralization where stimulation at the right branch of the vagus nerve basically has more of a motivational effect, whereas if you stimulate on the left side, it probably has a stronger effect in driving changes in the frequency of the stomach, for example. And it might be simply due to an asymmetry of the projection targets. So this is known that the left vagus nerve innervates slightly different targets in the nucleus of the solitary tract than the right vagus nerve. And similarly, it has been reported that innervation of peripheral organs is different for the left and the right side of the vagus nerve. So in some sense, there seems to be a kind of specialization, if you will. I don't know why that is, but I think it's reproducible across experiments that you don't really see a good dopaminergic signal if you stimulate the left nodose ganglion. Whereas if you stimulate the right nodus ganglion, you can get those motivational effects. And yeah, I don't know why that is, but it seems to be inherent in the circuit.
Thank you so much, Professor. One of our participants and club members named Efe wants to ask his question. Efe, you may now speak.
Initially, I want to thank you for your precious presentation. In your research, we observed that stimulation of vagus is via the auricular branch. And in microbiology, the flora of gut is strictly associated with the brain and the mood of the person. Can we find any other factor such as flora of the gut that affects brain via the vagus nerve? What if the flora changes? Does it affect the afferent vagal stimuli that leads to neurospasticity?
Yeah, so that's a great question. So there is a lot of good work showing that many of the modulatory effects of the microbiome are dependent on functional integrity of the vagus nerve, right? So if you do a vagotomy as well, you can abolish most of the effects of the microbiome on behavior and affect. And one of the things that is perhaps surprisingly not really known is whether, stimulation of vagal afferents also changes basically the effect of the microbiome on behavior, whether you can upregulate it or downregulate it if you use a different kind of stimulation protocol. That's something that hasn't been fully explored yet. If we can basically also regulate the weight that we put on signals from the gut by modulating, projections to the brain. I think that's also a very fascinating kind of topic. We were planning to look into this also in the future, but we are somewhat dependent also on animal research to do the more tricky kind of invasive work, because it's very difficult to isolate some of the components in humans as well. But yeah, we also don't know whether, for example, stimulation would change long-term composition of the microbiome and things like that. It hasn't been investigated in combination, and that's also because if you look at the animal research, it's often more acute invasive stimulation and less of chronic non-invasive stimulation that you use in humans. And I think if you want to know whether you could alter this, you would have to use some of those long-term stimulation protocols as well that are in human studies, and basically translate this to animals as well.
Thank you for your answer.
Thank you once again, Professor. One of our participants. Sorry.
Yeah, should I just go ahead and answer the second question? Should I read it again?
I was gonna read it out.
Okay.
But, okay, thanks.
You then go ahead. Sorry.
One of our participants asked, your study in Nature Communications showed that transcutaneous vagal nerve stimulation increased invigoration to work for rewards without altering effort maintenance. Can you explain why the distinction between invigoration and effort maintenance and these findings are significant?
Yes. So thanks for this question. We spend a bit of space also on separating the two components in the paper, but I didn't really elaborate on it in the talk. The main difference between invigoration and maintenance is invigoration is primarily driven by the potential benefit of your action, and it doesn't really take into account costs of effort, okay? So we showed that invigoration is mostly independent of the difficulty level. It has a small effect of difficulty on invigoration. But more importantly, I think invigoration is strongly associated with subjective ratings of wanting, but not with subjective ratings of exertion, whereas effort maintenance is associated with wanting and exertion. And effort maintenance is also more strongly affected by the difficulty of the task. So in other words, I think what is happening is invigoration is the sort of automatic approach or motivational drive that I mentioned, right? So you don't really think about it first. If you see a promising reward, you more or less go automatically to target this reward and try to collect it. And then you may update your effort on the fly, depending on whether you're still willing to invest the effort. And if it gets harder to maintain your behavior, you may just decide to take a break. Okay. And that cost-sensitive aspect is captured in effort maintenance, but it's not captured in invigoration. And with a similar kind of task, it has been shown that SSRIs, for example, so the conventional treatment for major depressive disorder, they improve effort maintenance by making healthy participants also more tolerable to the cost of effort. And that might be a mechanism why they also help in major depressive disorder. But it's important to keep in mind that even in major depressive disorder, they do not really treat symptoms of anhedonia. So they are very effective in improving symptoms of anxiety and mood, but not so much in treating motivational symptoms. And one of the reasons might be because they're not really affecting the reward component, but more the cost component of behavior. Okay.
Thank you, Professor, for your answer. Our participants Arda asked, overall, what do you think are the most significant implications of your study for understanding human motivation and the potential use of non-invasive brain stimulation techniques in modulating reward function?
Right. Well, we do hope that we could use non-invasive brain stimulation to help participants improve their reward-related behavior, and that's not necessarily restricted to conditions such as anhedonia. My vision is that it will be possible, let's say in 10 years from now, that people come to the lab and say, I want to change this sort of behavior, and that we know how we can stimulate the individual brain to kind of support relearning of behavior more broadly, right? So far, I think there are a lot of clinical applications where it is of interest, but I think when you think about deliberate weight loss as well in participants who are not necessarily obese, but only overweight, you are at a given zone where it's not clinically indicated, but there is a large also wish to just alter behavior, maybe reduce consumption of certain unhealthy food options. And we don't really have good tools to support this. So our hope is that we can develop non-invasive brain stimulation tools that can help us regain control over our behavior, in a sense that if we want to modify it, there is a way how we can use non-invasive brain stimulation to relearn certain behavioral routines. And that not only is related to food rewards, of course, but maybe also to addiction more broadly, right? Where we would like to just also break certain behavioral habits that are very difficult to target with conventional treatments, right? Other than, say, cognitive behavioral therapy. But yeah, that's sort of our vision. And we think that the gut is promising in that sense because we have a physiological mechanism, hunger, that is quick to upregulate motivation. And we also have an opponent system, related to satiety that could help us shut down reward-related behavior. So if we figure out how to recruit those circuits, how we make use of endogenous mechanisms to regulate reward function, I think we might be able to improve also the treatment of various conditions and maybe also help people improve certain habits that they want to get rid of. But that's, certainly the second step beyond treating some of the more important and debilitating clinical conditions.
Professor, thank you so much for your answers. I'm just going to wait for a couple of seconds if there are any new questions in the chat box. Okay, then I think we can wrap up. I really enjoyed the lecture, and I hope you enjoyed our seminar as well, Professor. Once again, I extend my heartfelt thanks to you for sharing your expertise and illuminating us with your groundbreaking research.
Thank you very much again for the invitation. It was my pleasure. And yeah, thank you very much for running this Neuroscience Society as well. I think it's always good to have that sort of initiative at the university. So my pleasure to support this.
Thank you so much. And thank you all for your participation. We hope to see you again in our upcoming events. Have a nice evening all.
Bye.